Good morning everyone. Today we'll continue the ongoing molecular biology lecture classes. Today we'll discuss about gene mutation and mechanism of regulation of gene expression, which is included in competency B17.3. Today we'll discuss about regulation of gene expression. So learning objectives. So at the end of this session, all of you should be able to explain that many steps involved in vectorial process of gene expression, which range from targeted modulation of gene copy number to gene rearrangement to transcription to messenger RNA processing and transport from the nucleus to translation to protein subcellular compartmentalization to post-translation modification and degradation, and these are all subject to regulatory control of both positive and negative. At the end of this session, everyone should be able to know the changes in any or multiple of this process can increase or decrease throughout the activity of the cognate gene product. And at the end of this session, everyone should be able to appreciate that DNA binding transcription factors, protein that bind to specific DNA sequences that are often locked near to transcriptional promoter element can either be activate or repress gene transcription. So at the end of this session, you all should be able to recognize that DNA binding transcription factor are often modular proteins that are composed of structure and functionally distinct domain which can directly or indirectly control messenger RNA gene transcription either through contact with RNA polymerase or its cofactors or through interaction with co-regulator that modulate nucleosome structure via covalent modifications, and nucleosome directed displacement typically increase or decrease. So now coming to the biochemical importance of today's topic. So biomedical importance. Organisms adapt to environmental changes, altering gene expression. Mechanism of controlling gene expression involve modulation, gene transcription, control transcription, which results from changes in the mode and interaction of specific regulatory molecules, proteins with various regions in the DNA in the control genes. And there are interactions, maybe positive or negative effect on transcription, and transcription control can result tissue specific gene expression, and gene regulation influence hormones, growth factors, heavy metals and chemicals. And also by transcription level control, gene expression can also be modulated, gene amplifications, gene rearrangements, post-transcriptional modifications, RNA control and protein modification, protein compartmentalization and protein stabilization. Also, why it is important of this gene regulation, many of the mechanisms that control gene expression use respond developmental cues, growth factors and hormonal as well as environmental agents and therapeutic drugs. By this regulation of this gene expression can lead to human disease. So the molecular understanding of this gene regulation or gene expression process lead to development of therapeutic agents, alter the pathophysiological mechanism which inhibit the function and arrest the growth of pathogenic organisms. So now coming to regulated expression of genes required for development, differentiation and adaptation. So genetic informations present in each normal somatic cell metazoan organism which is identical. Genetically reproducible hardware exceptions for in those few cells that have amplified or rearranged genes in order to perform specialized cellular fractions. Various disease state chromosome integrity which will turn into cancer. So this table shows the effect of positive and negative regulation of gene expression. When there is regulator is present, there is negative regulation, so it's decreased, and when regulator is present, in case of positive regulation, it will be increased. When there is absence of regulator, then negative regulation will be increased, whereas when there is regulatory regulator is absent, the positive regulation will be decreased. So when the expression of genetic information is diminished by the presence of specific regulatory element, which is known as negative regulation, an element or molecule mediating negative regulation or negative regulator or a silencer or a repressor. In case of mediating positive regulation, there are positive regulation. In this case, there is an enhancer or an activator, and maybe also there may be double negative, which will become a positive effect. An effector inhibits and function as negative regulator will appear to bring about positive regulation. So many regulated systems that appear to be induced are in fact de-repressed at the molecular level. So what is the biological systems which exhibit three types of temporal response to a number one regulatory signal type A. So this is how in type A regulatory signal how the genes are expressed, and type B, and here there is recovery time signal. Then type C, here this diagram represents the response of the extent of expression of a gene to a specific regulatory signal such as a hormone as a function of time. So in type A response is characterized by an increased extent of gene expression which is dependent upon the continued presence of an inducing signal, and this inducing signal remove the amount of the gene expression, diminishes to its basal level, which repeatedly increases response to the reappearance of a specific signal, commonly observed in prokaryotes, which responds by sudden changes in the intracellular concentration of the nutrient. A type is observed in higher organisms after exposure to some inducers like hormones, nutrients, or growth factor. And what happened to the type B response? Type B response exhibits increased amount of gene expression that transient even in the continued presence of regulatory signal. So this regulatory signal is terminated in the cell, then allowed to recover again. So there is a second transient response to a subsequent regulatory signal is observed. This phenomenon and response of desensitization recovery characterizes the action of many pharmacological agents, and these features may be naturally occurring processes. B type is commonly occur during the development of an organism when only the transient appearance of specific gene product is required, although the signal persists. And what happened to type C? In type C, the response pattern exhibits in response to the regulatory signal an increased extent of gene expression that persists indefinitely even after termination of the signal. The signal acts as a trigger. Once expression of the gene occurs, the gene is initiated in the cell. It cannot be terminated. Even daughter cells is irreversible and inherited alteration. This type C response typically occur during the development and differential function in a tissue or in an organ. So now coming to the gene expression, what we are going to discuss today, what is the mechanism of this gene expression. So gene expression is the process by which formation from a gene is used in the synthesis of a functional gene product, and these products are often proteins, but in non-protein coding genes such as ribosomal RNA genes or transfer RNA genes, the product is a functional RNA. So expression of gene expression regulation, here both of these cells contain the same genome but they express different RNAs and proteins. So now coming to the classification of gene with respect to their expression, may be conjugative or housekeeping genes. Number one, they are expressed at a fixed rate irrespective of the cell condition and their structure is simpler. And controllable gene, here these controllable genes are expressed only as needed. Their amount may increase or decrease with respect to their basal level in different conditions. Secondly, these controllable genes, their structure is relatively complicated with some response elements. So several steps in the gene expression process may be modulated, including number one, transcription, number two, RNA splicing, number three, translation, and finally post-translational modification of a protein. So process of alteration of gene expression has been studied in detail and involves modulation of gene transcription. So this gene transcription control can result in tissues specific gene expression and influenced by hormones or by various heavy metals. In simple terms, regulation of gene expression is of two types. Number one is positive regulation, number two negative regulation. So what is this positive regulation? When the expression of genetic is quantitatively increased by the presence of a specific regulatory element, it is known as positive regulation. The element modulating this positive regulation is known as an activator or a positive regulator. And in case of negative regulation, when the expression of genetic information diminishes by the presence of specific regulatory element, the element or the molecule mediating the negative regulation is said to be a repressor. So to know and explain regulation of bacterial gene expression, the constitutive or housekeeping versus controllable genes. There is operant structure and its role in gene regulation and regulation in eukaryotic gene. Then this expression of gene at different DNA methylation, increasing the number of gene copies, that is gene amplification, changing the rate of initiation of transcription, alternate splicing, messenger RNA stability, changing the rate of initiation of translation, using of untranslating region, protein stability, hormonal regulation, and there is also crosstalk between different regulatory pathways. So regulation of gene expression or gene regulation includes the process that cells and viruses use to turn the information in genes into gene products. Although a functional gene product may be an RNA or a protein, the majority of the known mechanisms regulate a protein coding genes. So any step of the gene expression may be modulated from DNA to RNA transcription, then to the post-translational modification of a protein. This is how the DNA is transcribed, then transcription, then splicing, then translation, then post-translational modification, and finally the finished protein. So gene regulation is essential for viruses, prokaryotes and eukaryotes as it increases the versatility and adaptability of an organism by allowing the cell to express protein when needed. So the first discovered example of a gene regulation system was the leg operon discovered by scientist Jack Monod, in which protein involved in lactose metabolism are expressed by E. coli only in the presence of lactose and absence of glucose. So furthermore, gene regulation drives the process of cellular differentiation and morphogenesis, leading to the creation of different cell types in multicellular organisms, where a different type of cells may possess different gene expression profile. So regulation of gene expression can be studied under these headings, that is the principle of gene regulation, regulation of gene expression in prokaryote, and regulation of gene expression in eukaryotes. So the principle of gene regulation. So what is it? Here most prokaryotic genes are regulated in units called operons, and these scientists Francois Jacob and J.Q. Mon in the year 1961. This is largely based on regulation of lactose metabolism. So this is the structure of an operon. You can see the DNA, then the regulatory sequences, the activator binding site, repressor binding site, the promoter, and these are the different genes to be transcribed as a unit. So principle of gene regulation. Number one, RNA polymerase binds to DNA at promoters. Then transcription initiation is regulated by protein that bind to or near promoter, and repression of a repressible gene, that is negative regulation, repressor versus activators bind to the operators of DNA, and repressor is regulated by an effector, usually small molecule or a protein that binds and cause a conformational change. An activator binds to DNA site called enhancer to enhance the RNA polymerase activity, becoming positive regulation and induction of an inducible gene, for example, heat shock genes. So this is how when a molecular signal causes dissociation of regulatory protein from DNA, and this is the example of negative regulation. You can see here the operator here, the promoter, and here the molecular signal, and then here this positive regulator. Here you can see the DNA RNA polymerase, here the product messenger RNA. And in case of molecular signal which cause binding of regulatory protein to DNA, and this is the product messenger RNA when the molecular signal causes binding of regulatory protein to the DNA. One is without regulatory protein where there is dissociation, and here there is binding of the regulatory protein.